Good morning, everyone. Like Alex mentioned, my name is Sam, and I'm going to be presenting my honors capstone, which is on abnormal behavioral phenotypes in juvenile and adult FMR1 knockout mice. I completed this capstone under Carrie Jonak and Dr. Binder in Binder Lab, which is located in the Biomedical Sciences Department of the UCR School of Medicine. So to give FMR1 some context, I want you to think back to elementary school or middle school. I want you to remember the person in your class that got scared and covered their ears every time the bell rang, or was frightened every single time the fire alarm would go off. And I mean every single time the noise would ring. They were probably the person you saw with the teacher's aide while they were covered in their ears trying to remain calm, or they were the ones that would come to school before or after the fire drills. I want you to remember if this person ever came up to you on the playground. Were they laughed at because they ran when the ball was thrown at them, or when someone touched them in tag? The reason why I want you to remember this person is because this person is why I'm going to spend the next 14-ish minutes talking about my project on abnormal behavioral phenotypes in juvenile and adult FMR1 knockout mice. So what is FMR1? FMR1 is also referred to as the Fragile X messenger ribonucleoprotein 1 gene. And essentially, it's the gene responsible for Fragile X syndrome, which is the most common genetic cause of intellectual disability. Approximately 1 in 4,000 males and 1 in 6,000 to 8,000 females possess Fragile X syndrome. So this mutation is located here on the X chromosome, right here. And it consists of a cytosine guanine guanine repeat, or a CGG repeat, which is pictured over here. This essentially means that these base pairs continuously repeat, and if you get the sequence 200 or more times, the mutation is present. Anything less than that, it can be a carrier. And this leads to the loss of the Fragile X messenger ribonucleoprotein known as FMRP. FMRP is a RNA binding protein, so this is what RNA looks like here, that regulates brain function. So if FMRP cannot bind to the RNA strand, this makes it so their brain function goes unregulated, and this is what occurs in Fragile X. This can lead to symptoms such as increased anxiety, repetitive behaviors, abnormal sensory processing, and more. So essentially, what does Fragile X look like? So over here, you can see my little P21s, which are 21-day-old mice, and they're the age equivalent of young children. Over here, you can see children with Fragile X syndrome. Some of the similarities are the enlarged ears and the long snouts or the long faces. In children, this can be presented as shyness, repetitive or rapid speech, anxiety, or difficulty making eye contact. In mice, it presents as a sense that they don't really want to be around others. They don't tend to build their nest. They like to dig a lot, and they're very fidgety and anxious. So what is the point of exploring this? It has never been done in juvenile mice, which when we treat Fragile X in clinical studies, it's generally done in children. So by doing this experiment, it allows us to create a better age-related comparison versus just focusing on adult mice, and allows a more targeted translational model. It also allows us to do treatments more holistically versus just looking on the neurochemistry side. So my study consisted of a nesting behavior assay, which was 2 different types. We did a nesting removal or digging, and we did nest building. The reason why we did nesting removal was because people that have Fragile X or autism-related disorders like to maintain a very specific routine and environment, and the digging test in mice replicates this idea, and also takes it one step further with us agitating their environment. When we agitate their environment, it disrupts their normalcy, which then creates more and higher stress levels and anxiety-related behaviors. We also did a nest building test for mice, and that's because rodents have this innate or the natural want to create a nest. And so this plays into their sensory processing, because in Fragile X, there's an abnormal sensory processing, which means that they would have a lesser desire to actually build these nests. So for the nesting removal, we allowed the mice to stay in their cage undisturbed for 3 days, this is what the cages look like. They also had access to food and water whenever they wanted through a feeder, and they were kept on a 12-hour light cycle. So 12 hours at night was dark, and then 12 hours during the day with light. On the day of the test, we took them to the room and let them sit and habituate, so essentially just get used to the room for an hour before we started. Then we proceeded to record them from above, which this is the view of the cameras, for 10 minutes undisturbed. So this is what the undisturbed looks like. Then we removed their nest, creating that agitation, and recorded it. So as you can see here, you can see him plowing and digging through. This is when we recorded it with a stopwatch. And so every single time they did a movement like that, or we're just digging like you're playing in a sandbox, we stopped and started the stopwatch. And that is how we recorded the amount of time they spent digging. We did this with both the juvenile and adult mice. We had 15 of each, so 15 possessing Fragile X, which is the KO, and 15 not having Fragile X, which is the WT, so wild type. So here you can see both in adult males and P21 males, when they have their nest, there is no difference or no significant difference between the amount of digging they do, which is what we expect because we're not disrupting their environment and therefore creating or inducing anxiety. When we do, however, remove the nestlets from their cages, you can see how there's an increase in anxiety in both males or adults and P21s. Our p-values were assessed at 0.05, with the major significance here being 0.01 and the major significance here being 0.1. You can tell that there's an increase in anxiety, and this is what these diagrams are showing. So then we moved on to the nesting or building the nest. Both the adults and P21 mice followed the same protocol, except for an adjustment in the size of the nestlet, which I will talk about in a minute. So they were originally housed by themselves, and they were given their nestlet 1 hour before dark cycle, so approximately 6 p.m. They were then given overnight to make their nest, and it was removed at 8 a.m. the next morning. First, we took photographs of the nest, so this is what they look like the next morning, and then removed them and went through the nest. So we were just pulling them apart, finding the chunks that were still intact. The largest piece that was intact was then used to create the score because that represented essentially the nestlet that they did not rip apart. They were scored by according to Deacon's protocol, and it's on a 5-point scale. Here's some images down here that represent each, A corresponding to 1, B2, C3, D4, and E5. They were assessed based off of visual, so how high the nest was around it, how well it was distributed in the cage, as well as the percent remaining intact. And that was taken by weighing the nestlet before given and then weighing the nestlet after. And you can see here in the adult mice that the wild type had the want to build their nestlet or their nest more so than the mice that possessed Fragile X, which shows that they don't have that same sensory processing deficit as the Fragile X mice do. So because Fragile X mice have that sensory processing deficit, it makes them not necessarily understand what to do with the nestlet as readily as the mice that do not have Fragile X. You can see over here with the nestlet intact, it reinforces that idea because Fragile X mice have a larger percentage of their nestlet remaining even after being given the same amount of time to create their nest. However, because the P21s are so small, so adult mice range around 30 to 35 grams, the P21s range about 8 to 10 grams. So you can see in comparison to the adult size nestlet, they're very, very small. And so they did not touch the nestlet, neither wild type nor knockout mice did. So then we assessed the nestlet to 9% of the P21's weight instead of the standard 9% of the adult's weight. and it was still too large. From there, pictured here, we assessed it to about 7%, so anywhere between 0.8 to 0.7 grams. The wild types did start interacting with the nestlet, however, their scores were really low. So we dropped down the weight of the nestlet to about 4.5 to 5, which you can see here, it's definitely ripped up, which is approximately 0.5 grams. And this is what we used to do the nest building test for the P21s. They followed the same exact protocol as the adults did and yielded the same results. So like the adults, the P21 wild types had the want to build their nest more so than the knockout mice, as well as the knockout mice had more of their nestlet remaining the next morning in comparison to the wild type mice. So what does this all mean in terms of this study and essentially the bigger picture? So this showed that both adult males and P21s have that same abnormal behavioral phenotype. And this allows us to create a baseline for future drug studies, as well as create a more holistic view when we're doing our collaborations with Cincinnati's Children's Hospital to ensure that our models are better representations of the children's age group that we collaborate with. I would like to acknowledge my entire lab for helping me throughout this entire process. So over here is pictured my lab partner, Mambir, our grad student, Ali, who is doing her dissertation in a couple weeks, and our lab assistant, Therese. Here is Dr. Binder and my lab manager, Carrie. This is us pictured at our neuro conference the year before the pandemic started. And then these are all of their lovely headshots. Shreya and Patty, as well as Manolia. And I want to give a special thanks to Therese and Manolia and Carrie, because they have listened to me go through this entire thing and helped me so much revising everything and being here telling me I can do it. But thank you for listening to my presentation.
Let's thank Sam for that great presentation. Great job. So we have time for questions. Would anybody like to ask a question? You can also ask questions in the chat if you can't or don't want to use your microphone.
All right, that's a really wonderful talk, and I think such a clear experiment, and really great applications. So thank you for telling us about it. I was curious, in some of the data you had, you presented it as a bar, and the error bars or the whatever those were, right? I was just curious, did you see a sort of a monotonic distribution of behaviors, or did you see subpopulations of your mice that were giving a different response, right? Because the width of the distribution seemed quite big. And is that just coming from variation on a single phenotype, or is it that there are clusters of them that give you slightly different behaviors?
Yeah, so it's essentially just over across all of them. So there are mice here and there because our N of 15 is it encompasses all of the phenotypes in the sense of that, but it is that variation amongst, because sometimes maybe we caught one on an off day or they were just being mean to us that day. But it is that variation just amongst each other. But it's generally consistent with that significance that they are all still in a deficit regardless of that variation.
So it's a very nice analysis, and thank you again for your talk.
Great. Other questions? Don't all jump in at once.
Yes, I do have a question for Sam. You did awesome, by the way. Your presentation was super spot on. Yes, I do have a question. So you had mentioned earlier in your presentation that children with Fragile X exhibit behavior where they kind of like want to be isolated and by themselves, right? Would you expect that same phenotype if we were to group house the mice instead of single housing them on the day of the experiment, or what would you expect?
So I'm glad you asked that because we've actually experienced a little bit of both, where they kind of want nothing to do with each other. But then when we started doing per se like surgeries, they were very like tight knit and huddled together, making sure everyone was okay. So I want to say it's more dependent on what you're doing with them, how agitated, like with one of the experiments, you leave them for 3 days. So leaving them for 3 days, they tend to calm down a little more. Plus, generally when you group house, they're litter mates. So they were born together and they're already together and they're familiar. But if you were to separate them and then regroup house them, I would believe that they would not like each other. That's when you start getting your fights. But I'm not 100% certain.
Great answer. Thank you.
Do you have time for more questions? Well, while some folks might think a little bit more about questions, I do have one. I thought it was really interesting, the behavior and the nesting response. So it seemed like some of the P21s were hesitant to take on the larger nestlet. Do you think it was fear or just being overwhelmed, or because I think there's parallels to
I think it was a little more so on overwhelmed, as well as this was more so like their first introduction to something like this because we just weaned them from their parents. So it's that idea of, okay, you have this giant thing, what do I do with it? It's the idea of being a little bit scared and overwhelmed with that because of the fact that there is this sensory processing deficit. So even when we did decrease it to a more manageable size, that's when we started having the wild types actually interact with it because they don't possess Fragile X. But then when we started to getting it down to a more manageable size for both phenotypes, that's when it was very clear that they were still not really understanding what they were processing and what to do with the materials, even though it is a natural instinct to create a nest.
Yeah, thank you for that.
I have one last question for Ms. Sam.
Yes, go ahead.
So with behavior studies, typically there's a lot of question around bias and how you stay blinded to the study, and if the study is, remaining true and remaining blinded. So how did you guys deal with that in this experiment specifically?
Yeah, so I was the one that was doing all of the timing. So the digging analysis was the same across the board because I was the one constantly doing the recordings on that. As far as staying blinded to the phenotypes or which mice I was dealing with that day, it was you, Mambir, and Carrie, depending on what was going on in that sense, where the cage cards were changed colors. I was told to stand outside while everything was sorted. Okay, now you can come in and run it. They were given numbers or just different like letter groups depending on which ones we were doing that day. So it was all very randomized, and I didn't know which ones belonged to which until the very end when everything was collected and the data was being run.
Awesome, thank you.
Great. I think I don't see any more questions. So if that is the case, I think we're going to be moving on to our next presentation. But let's thank Sam once again. Sam, great to see you, and best of luck to you with the rest of the year and your future studies.
Thank you.